Let's take a look at nucleosides, nucleotides, and nucleic acid. So first, let's take a look at nucleosides. Molecules containing sugar moiety and non-containing base moiety are nucleosides. In RNA, the monosaccharide is aldopentose derivose. This is derivose here. This is a 5 membrane, 4 carbon atoms, and 1 oxygen. 1, 2, 3, 4. The C5 is here. In DNA, the monosaccharide is D2 deoxyribose. Now, although pentose that like hydroxy group at the C2 is the C2 position here, and you can see the hydroxy group is missing at C2. This is D2 deoxyribose. Only 5 common nitrogen-containing bases are present in nucleic acid. 3 bases with 1 ring, cytosine, uracil, and thymine, are derived from the parent compound called pyrimidine. 2 bases with 2 rings, adenine and guanine, are derived from the parent compound called purine. Each base is designated by 1 letter abbreviation. Cytosine is C, uracil is U, and thymine is T, adenine is A, and guanine is G. Let's take a look at those structures. It's pyrimidine is here is 6 membrane, 1, 2, 3, 4, 5, 6. It's a cytosine. Cytosine has a carbonyl group at the C2 position and amino group at the C4 position. Uracil has a carbonyl group at the C2 and another carbonyl group at the C4. Thymine is different from uracil by having a methyl group at the C5. This is C5 here. This is purine. This is a 2-ring compound. Adenine has amino group at the C4. This is a number 1, 2, 3, 4, 5, 6, 7, 8, 9. The guanine guanine has amino group at the C2 and the carbonyl group at the C4. A nucleoside is formed by joining the anomeric carbon of a monosaccharide with the nitrogen atom of the base. Nucleoside is a non-nucleoside because it's a nitrogen derivative of the glycoside. Primers, the sign of this. The primes are used to number the carbons of a monosaccharide in a nucleoside to distinguish them from the carbon atoms in the ring of the base. With the pyrimidine bases, the nitrogen atom at the 1 position bonds with the 1' carbon of the sugar. 1' carbon also is called the anomeric carbon. With the purine bases, the nitrogen atom at the 9 position bonds with the 1' carbon of the sugar. This is the structure information. Let's take a look at this. This is ribose. This is cytosine. And this is called cytidine. Cytidine is the nucleoside. This is the base, cytosine. You can see this is the C1 position of the sugar. This is the N1 position. They form the bond between C1 and N1. This is the new bond here. This next is adenine base 2-ring compound joined with the 2-deoxyribose. This is the C1 prime. This is the C9. This is the N9. A9 position. Then they form the nucleoside by forming the bond between C1 position of the sugar and A9 of the base. And this is called deoxy adenosine. Deoxyadenosine. How to name the nucleosides? There's some rules here. To name a nucleoside derived from pyrimidine base using suffix -ade. Example is cytosine becomes cytidine. To name nucleoside derived from purine base using suffix -osine. This example, adenine become adenosine. For deoxyribonucleosides, as the prefix deoxy, as in deoxyadenosine. Next, let's take a look at the nucleotides. Nucleotides are formed by adding a phosphate group to the 5' hydroxy group of a nucleoside. Ribonucleotides are derived from ribose, while deoxyribonucleotides are derived from deoxyribose. Nucleotides are named by adding the term 5' monophosphate to the name of the nucleoside from which they are derived. Abbreviations are used for nucleotides. For example, cytidine 5' monophosphate is abbreviated as CMP, and deoxyadenosine 5' monophosphate is DAMP. Let's take a look at the structural information. You can see this is a phosphate group. Phosphate is a cytidine. This is a nucleoside. This is a ribonucleoside. Then they form a new bond between phosphate and the C5 hydroxy group here. C5. This is a new bond here. This is the bond. It's a nucleotide. This nucleotide called cytidine 5' monophosphate. This is another example here. It's a phosphate. It's a deoxyribonucleoside. It's a deoxyadenosine. It's a two-ring compound. This is a C5' carbon. They form the bond with the phosphate, and this is a new bond. This nucleotide is called deoxyadenosine 5' monophosphate. Let's take a look at nucleosides, DNA and RNAs. Nucleosides are single chains of polymers of repeating monomers, nucleotides. There are two types of nucleic acids. The first one is DNA, is a deoxyribonucleoside, cytogenetic information of the organism. The second one is called RNA, ribonucleoside, with the major functions of transmitting genetic information for protein synthesis, among many other functions. Other functions including ribosome, it's the enzymes catalyzing some chemical functions, and macroRNAs. Nucleic acids are formed by joining 3' hydroxy group of one nucleotide with the 5' phosphate group of the second nucleotide in diphosphoester linkage. This is how this is polymerized. This is a 5' and has a phosphate group. This nucleotide is 3' and form the bond here and linked to the second nucleotide. And this is another one, and another one, and all the way to the 3' end. At the 3' end has a free hydroxy group. Briefly, let's take a look at some characteristics of nucleic acids. The first one, polynucleotides contain a backbone consisting of alternating sugar and phosphate groups. Number 2, identity and order of the bases distinguish one nucleotide from another. Number 3, polynucleotide has one free phosphate group at the C' end and a free hydroxy group at the 3' end. Number 4, one of the difference between DNA and RNA is that the sugar molecules in DNA is D2 deoxyribose, which lack hydroxy group at the C2 position, while the sugar molecule in RNA is D-ribose. Number 5, another difference between DNA and RNA is that DNA contains thymine and RNA contains uracil in addition to 3 common bases, adenine, guanine, and cytosine. In summary, DNA contains bases A, G, C, and T. RNA contains the bases A, G, C, and U.